Hey guys, welcome back. So now we're going to look at an example of the endomembrane system as it relates to sort of current events in medicine. So in the previous videos, we learned more about the general structure of the endomembrane system. We learned about the smooth ER, the rough ER, and how ribosomes know whether to translate into the rough ER. We also learned about the Golgi apparatus and how it can add and modify things like proteins, and it can actually sort them, sort of like a post office. So in this video, we'll look at why should we care about the endomembrane system. Okay, so why should we care? Our example today will be using SARS-CoV-2, aka coronavirus. So this is the general structure of a coronavirus. We've got the lipid envelope. So this is lipids just similar to the way that we have a plasma membrane. Spike proteins. There are other types of proteins that are present in the envelope of these viruses, but spike proteins are the ones that get the most attention because, as we'll see, they'll be the ones largely responsible for allowing this to get into human cells. We also have genetic material, in this case, RNA. So to understand a little bit more about how this relates to the Golgi apparatus and glycosylation, we need to know more about how viruses replicate in general. So viruses have no ability to replicate on their own. So SARS-CoV-2 must enter a host cell like those found in a human respiratory system to take advantage of the cell's machinery. So again, now we're just looking at the way that a virus replicates. So we'll start out with how it gets into a cell. So to enter the cell, the SARS-CoV-2 spike proteins must successfully interact with the receptor on our own cells. In this case, this is the Ace2 receptor. So this is why you hear about Ace2 getting a lot of attention in the context of coronavirus. So this is the receptor that facilitates entry, and it's present not only in the respiratory system, but in many cell types throughout the body, Ace2 receptor. Okay, so here we go, those spike proteins ready to interact with this receptor. Once the virus is docked on the receptor, an event called endocytosis is triggered, which means that the cell begins to bring its virus into the interior. Okay, so here is the vesicle forming to take the virus inside. This should maybe remind you of something. So we've got our vesicle, we've got our cytosol. Once inside, the virus has some work to do. Okay, so it's going to, again, try to utilize host machinery to replicate. So first, it dumps its RNA into the host cell cytoplasm. So you can see that the lipid envelope is going to actually end up merging with the endocytotic vesicle that took it in, so that we kind of inside out this envelope and dump the RNA into the host cell. Okay, so here's the RNA kind of stretched out. The unknowing host cell then replicates the RNA and uses it to make viral proteins. Okay, so here's ribosomes, our ribosomes. The virus doesn't have any of its own, so it uses our ribosomes to make more spike proteins, more proteins that are necessary, and eventually it will also make more copies of RNA so that it can package it. Okay, so some of these proteins, again, that it's making include the SARS-CoV-2 spike protein, which we saw was embedded in the virus lipid envelope. Because it spans a membrane, it must take the usual route through the ER and Golgi apparatus. Okay, so what happens in the Golgi? Okay, we remember that in the Golgi, we add things like carbohydrates. This is called glycosylation. Okay. Once all the components are made, virus particles are packaged and sent out of the cell by exocytosis. Okay, but now we know a little bit more about this, and that means that spike proteins are not just naked, but they actually have sugars attached. Okay, so this has sugars attached to our spike proteins. We've glycosylated them. This is a not so sweet surprise. Okay, so I want to show you guys a picture from a recent publication that actually mapped the positions of glycosylation in linked glycans on the spike protein. So this is the top view of the spike protein as if you're looking down into it. And then this is a side view. And you don't have to take a lot away from this, but basically, sort of all of these colored regions that are sort of these like magenta, green, orange, not the blue ones, but the other three kind of represent different glycosylations that are predicted to occur on the top and side of the spike protein. So you can see this is actually very heavily decorated with carbohydrates. And these glycans can perform several functions. But one function that researchers think is that these glycans actually help shield the virus from detection by the human immune system. So in principle, we would normally create and make antibodies to things like the spike protein. Okay, and this really, it's simple. It doesn't capture really what's going on when we make antibodies, but you get the idea. And this process is sort of inhibited when we have too many of these carbohydrates, making it more difficult to detect and get rid of through the immune system. So therefore, it's really important for research scientists and physicians to understand the way that viral proteins are structured. So you may have watched the first 3 videos thinking, I'll never need to know about the Golgi apparatus because I'm going to medical school. But again, it's really important to understand this because understanding the actual structure plus the carbohydrates that are added can help develop better treatments for vaccines, other viral illnesses.